Hello, my name is Floryanzia, and like he said, today I'm going to tell you a little bit about how chemical engineers are developing tools to explore the brain drug delivery terrain, ultimately to treat brain diseases. So let's dive right in. So you may be aware that the brain is a complex environment. All of your thoughts and feelings, dreaming, kicking a ball, you looking at me, processing everything that I'm saying right now, your brain is hard at work. And to do all the things that your brain has to do 24/7 every single day at the rate that they have to be done, your brain demands a lot of energy, namely in the form of oxygen and glucose. And so for an organ that's only about 2% of the adult body weight, it receives about 15% of the nutrients that you supply to your body, which is really, really wild. And so how does your brain that has this enormous energy supply meet that energy supply? Well, through about 400 miles of vasculature, vessels that run all the way through your brain. And so this is really great. You have this efficient system that supplies your oxygen and glucose, all your nutrients, to your brain through your blood. However, for the brain, blood can be a double-edged sword. So in addition to the good things that it supplies, it also can unfortunately have bacteria or toxins or pathogens or things that can mean the brain harm. And frankly, your brain does not have time to fight infections or do all these things and also keep your body going, keep you going at the rate that it has to be done. And also you as a person, like when I have a lot to get done, it's really difficult to do that if I'm working at a cluttered desk, right? So your neurons are really trying to send messages to each other, and if the environment that they're doing that is cluttered, they can't. And so your blood-brain barrier is here to be a filter, to let in certain things, but try to keep out these bad things. So yeah, fortunately, your brain has a nice system to answer this problem, called, as I mentioned, the blood-brain barrier. And so what does this look like, this blood-brain barrier, this barrier that protects the brain from things in the blood-brain barrier? So if we actually zoom in to one of the vessels in our brain, we see a little guy, a little cell guy, kind of holding on to this capillary. So a capillary, if you think of a tree as a vessel, right? You have a tree and it goes off into branches and those branches into twigs. So a capillary is a twig. It's the smallest vessel that branches off. So if you zoom into one of those capillaries, you see a cell kind of surrounding it. Now, if you zoom in one more time to the cross-section of that capillary, you actually see some other characters, a bunch of different other cells that together in the brain line the system. And so great, we have a barrier system. We have a way to regulate nutrient entry and also protect against toxins. So again, we are able to let in nutrients and oxygen. They go from the blood through those cells into the brain. And medicines? Well, that is not quite so easy. So in an effort to protect our brain from everything, our blood-brain barrier protects our brain from everything, which includes the medicines that we're trying to get in. And so you have small molecules like oxygen, glucose, alcohol, caffeine. We know these things get in. They're really small. They're relatively simplistic chemical structures, which is great. But if we're thinking about treating diseases like stroke or diseases like Alzheimer's or Parkinson's, even trying to increase the options for psychiatric drugs and developing targeted cancer treatments, well, these things can't be treated with those small molecules. These things need larger proteins or antibodies, which don't get through the blood-brain barrier. So when we're trying to design medicine for the brain, it turns out we're going to need some tools. So this is our little handy-dandy toolbox. You'll see him appear throughout the presentation to kind of be our landmark as we explore the tools that we're using to treat brain diseases. So the first in our toolbox arsenal is actually an understanding of the blood-brain barrier. Know thy enemy, if you will. Or in this case, I don't see it as an enemy, but I'm getting ahead of myself. So if we revisit our cross-section of the capillary, first we have endothelial cells, which are our main characters. And so these endothelial cells actually line the inside of capillaries all over our body. And this is what they typically look like. So they're beside each other, but there's a little bit of space, that gap there. And in that space, everywhere else in the body, that's kind of where medicine molecules can actually get through. But in the brain, this gap doesn't exist. And not only is there not a gap, but there's additional tight junction proteins. So these cells really are together. They do not want anything setting foot on their brain turf. These are the bouncers of the brain, which is why they're kind of the main character. And I am a chemical engineer, as the bio stated, and you might be wondering, what is a chemical engineer doing talking about neuroscience? Well, the endothelial cell is the favorite cell of a chemical engineer because they also appreciate fluid flow. And so they have these little projections, which are called, they're called cilia, these little guys. And so they actually can detect changes in the blood that's flowing, and then those changes actually translate to functions, which is really, really cool, and something we'll keep in mind for later. But yeah, so these are our endothelial cells, main characters, first line of defense in our blood-brain barrier. And then we have pericytes. They're our supportive best friends. So if we kind of think about the blood-brain barrier dynamics as like your favorite show or high school, these are the side characters. And so we don't actually know a lot about pericytes outside of how they directly impact endothelial cells. Because again, historically, endothelial cells have been the main character. So at least in the research field, these are not quite yet three-dimensional characters per se. They are typically kind of only relevant in how they interact with endothelial cells. So if we go back to our high school example, they say, oh yeah, we heard so-and-so been talking about you. And the endothelial says, okay, and so they kind of sense things in their surroundings and then pass those messages in the form of molecular signals to endothelial cells to help them modulate. And also, beautiful structure and function comes into play here too. They actually wrap around the capillaries and provide another layer in this barrier and also can kind of make things tighter or looser in response to environmental cues. And then after this layer of cells, we have, wait for it, another layer of cells. These are our astrocytes, or our prom queens, if you will. And so they provide their branches, again, structure and function, these branches are for a purpose. So on one side, one of their branches, their end feet, they actually wrap around the capillaries, again, providing another layer of structural support. And then another branch, they actually connect with neurons. And they say, hey, neurons, what's going on? Oh, we need more water? Okay, let more water in. Oh, okay, keep that out. Okay, so they just really want everybody to be friends. So this is not the mean prom queen. This is really the prom queen that everybody knows, everybody wants to be friends, and also the astrocytes want everybody to be friends. So they connect the neurons and the rest of the brain to the vessel system and the blood. And great, like I said, we have a nice, happy system that lets in our nutrients and oxygen, but not the medicines. So we have to get to that part. Okay, so it sounds like it's time for another tool. And so in addition to having the tool of knowing the characters of the blood-brain barrier, we also have another tool called nanoparticles. And so you may be familiar with the concept, a little bit of nanoparticles, the same technology in the COVID-19 vaccine. Basically, this is a way to carry medicine, a new way to be a vehicle to actually take these nanoparticles, these medicine molecules into the brain. And so this is teeny tiny packages of medicine to kind of be more similar to the caffeine and alcohol molecules I talked about earlier, something that the brain might know. And when I said teeny tiny, I mean teeny tiny. So a human hair is about 100,000 nanometers, and our nanoparticles are about 100 nanometers. So a human hair is about the width of 1,000 nanoparticles, and I can't even see the width of a human hair. So teeny, teeny, tiny packages. And in addition to being very small, we can also change a lot about the properties of these nanoparticles. They can change their shape or their squishiness. We call it stiffness, but it's really just how squishy they are. And they can do something called switching, which is when they actually sense that they're in the cell, they can actually take off some of their different properties to better target some cells and avoid others. And by avoid, I mean the white blood cells who were like, who are you? You're not supposed to be here, right? But we do want them to get into the brain cells. So we need to evade some, be uptaken by others. And another way to do this is actually by add-ons. So we can put on different proteins or antibodies to kind of disguise our nanoparticles. Oh, nothing to see here. Bouncers, I am just a simple sugar molecule. Of course, you know me, you can let me by. So we can kind of have these add-ons to help the brain, the blood-brain barrier, want to accept the nanoparticles. And the good news is, we know this works. Our lab has shown that nanoparticles are able to get through the blood-brain barrier and deep within the brain. So great, we've cured all the brain diseases. Not so fast there. So we know that they do get in, but the question remains, how? And who exactly are these nanoparticles interacting with? What cells are allowing these nanoparticles to get in? Why are they being allowed to get in? So it's like we know that it works, but if we don't actually know why, then we're just still throwing stuff against the wall and hoping something will stick. And so the drug development process is still kind of halted. So it sounds like we need another tool. So the third tool that I'll be talking about today, and that's really the bulk of my research and the work that I'm doing as a PhD student, is called engineered model systems. And so if we take our little lovely brain, there's actually several ways that we can look at it. And so one is like tissue slices. So if you think of like a loaf of bread, we can actually slice a brain and look at the individual brain loaf slices, if you will. And another way is with the individual cells themselves. So I'll get into each one of these. And so if we take our tissue slices, this is actually one of the slices that are in our lab, and this is what it looks like. And so you can kind of see some little specks there. So if we zoom in, you can actually see the different vessels that are flowing through this brain slice. And if we zoom in again, you can see individual capillaries built with those individual endothelial cells, which is really, really cool to see. We also have our prom queen astrocytes. They're doing what they do. They're connecting with everybody in the brain with their long branches. But it's a little difficult to see exactly what's going on. Even though it's only a slice of the brain, there's still a lot going on. And so teasing out individual cell contributions or specific things that are happening is a little bit more difficult in the slice. So sometimes we like to split the brain up into the individual cell types. And so, again, here's our endothelial cells, but instead of being in their vessel shape, they're laid out flat. And so we can actually see the different widths of the different cells, the different lengths of the different cells, which is a little bit more difficult to see when they're wrapped up in the tube. And we also have our pericytes, our supportive best friends, make a reentrance. When they're in the slice, because they just wrap around the vessels, they are almost indistinguishable from the endothelial cells. But when we take them in isolation and see them, we see them spread out, and I get a chance to get a little bit more information that way. And then we have our prom queen astrocytes again. They're doing their thing, making their connections, but it's a lot clearer to see. We can see how thick are the branches, how big are the cell bodies, and those types of things actually change in disease. So it's kind of good to be able to see. And so in my work, I wanted to put an end to the main character-only way of looking at the blood-brain barrier. So I actually focus on the supportive best friends and the prom queen cells in the work that I do. And so taking these cells, I do something else. I actually put them in this handheld small microfluidic device. So microfluidic, micro, small, fluidic flow chamber. So this is what it looks like if we were to zoom in. You have this little channel. So you have flow that flows through the red channels, and it kind of allows us to make a vessel so that we can actually look at how the different nanoparticles actually interact with the different cells that make up the vessels of the brain. And so again, if we zoom in that microfluidic chip even more, we see that we can actually have the endothelial cells grow along the outside, lining the inside of that channel, and then we have the other cells that are kind of suspended and growing in 3 dimensions. And so in this way, it kind of resembles our blood-brain barrier. We have a layer of endothelial cells that are surrounding the outside, and then that's immediately next to the pericytes and astrocytes and the other cells of the brain. And so this is a real image of some of those actual cells. You can see that they're expanding in their environment, and we actually flow fluid through those channels. So we have the nanoparticles, we suspend them in solution, and that's kind of like nanoparticles in the blood, and then how they actually interact with the different cells. So we have a little tiny window to view brain cells and drug uptake in the manner that they're kind of like in vivo. So remember I talked about endothelial cells love flow. They have the little cilia. They need that flow to actually function properly as endothelial cells. So as chemical engineers, we use these microfluidic devices to bring in that element of fluid flow in our model systems so that our model systems actually give us accurate data when we go to test our different medicines. And so this is kind of what some of what I look at. This is the results of those experiments. So I have the little cartoons just to be your reference. These are astrocytes in the green and the pericytes in the purple, and all those red dots are actually the nanoparticles. So I guess this is a good way to actually see the difference in size too. These are the 100 nanometer nanoparticles. And so we see that some of the nanoparticles are associating around the nucleus of a lot of cells. Those are those blue dots. But it doesn't really appear that the nanoparticles are surrounding as much with the astrocytes. And so why does this matter? Well, astrocytes are being implicated more and more every day in diseases such as Alzheimer's. And so even if we get the nanoparticles through the blood-brain barrier, if they're not entering the cells that we want them to, then we need to go back to the drawing board and kind of edit our drug delivery and our drug designs. Similarly, our pericytes, they're implicated in a lot of different things. They are moving out of their supportive best friend role, and new research is coming every day. Huntington's disease, also Alzheimer's disease, and also in stroke. They're a target for repairing the brain after stroke. And so we need to know, are our nanoparticles able to get in to these pericytes? And do the pericytes even want them there? And so this is another view actually in the microfluidic channel. So you see that pericyte growing along and aligning to the vessel, which is, well, the microfluidic vessel, the vessel outside of the brain that we can use as our window. And so we see the nanoparticles in this one are actually inside one of our cells, which is great. So now we know that this nanoparticle can successfully get into this astrocyte, and thus, if we load a medicine into that nanoparticle package, we know it's going to get to astrocytes, and maybe it'll help with diseases such as Alzheimer's. And so yeah, finally, our toolbox again. With an understanding of the blood-brain barrier, with nanoparticles as packages for drug delivery, and with a knowledge to be able to see how these nanoparticles are interacting with the cells of the blood-brain barrier through our engineered model systems, we have the tools to deliver medicine to the brain, and thus the tools to treat brain diseases. Thank you.